Okay, now we are going to focus on meiosis. In meiosis, the cell progresses through the G1, S, and G2 phases of the cell cycle, where DNA is typically replicated. Here I am showing the prophase 1 of meiosis. So during prophase 1, the chromosomes condense and the homologous chromosomes pair up into tetrads, which is why they are right next to each other or on top of each other. During this stage, something unique happens. It's called crossing over between the homologous chromosomes. So when this happens, as you can see on my extra one, there is a part of the blue that actually goes onto the pink chromosome, which is what the crossing over is called, and that's signified by them crossing over at their touching points. Over here I have the centromeres and the spindle fibers as well. Now in metaphase 1, I still have my centromeres. During metaphase 1, the tetrads all line up on the metaphase plane, and as you can see, the spindle fibers are actually attached to the kinetochore from one pole of the chromosome to the centromere. Now, as I lay them down, there is something called independent assortment, and that states that I don't need, cells don't need to have the chromosomes lined up in a specific order. So as you can see, as long as they are still paired up, the two blue can go on the top, the two pink can go on the top, the blue can go on the bottom. That independent assortment makes that gene more unique as it progresses through the rest of the phases. So as you can see, one set of sister chromatids are attached to the south pole of the southern centromere, and one is attached to the northern centromere. Now going into anaphase 2, the spindle fibers are actually pulling the chromosomes to the opposite poles, and the sister chromatomes remain attached at the centromere. Also during anaphase 1 is when the cell starts to elongate and create that egg-like shape. Now in telophase and cytokinesis, the chromosomes the cell divides into two haploid cells, okay, with two sister chromatids. I'm gonna set this up while I talk about their differences. So with their differences, they both actually start between meiosis and mitosis. They start with a sister cell. That is a given they start with a diploid cell. That's a given, so sorry about that. But in meiosis, two cell divisions occur, which creates, in the end, 4 haploid cells. That fell apart. And then with each gamete can have a unique combination of genetics. So as you can see, the spindle fibers disappear. The nuclear envelope, which is my orange, starts to reform. Need to add a couple more in there. As you can see in here, they are different. So they are not the exact same. With that, then they split apart, and now we are actually in prophase 2. So another unique thing about meiosis, which is different from mitosis, is that meiosis, the cycles and the phases happen twice. So we are in prophase 2. In this phase, the spindle fibers reform. And then the chromosomes recondense, so they actually get a little bit closer to each other. Now, moving into metaphase 2. So the chromosomes are back on that metaphase plane. The nuclear envelope goes away. We do have our spindle fibers, so each of these is set up like so. Their spindle fibers, again, attached from the north and the south. I'm referring to it as north and south just because it's up and down. That is the cardinal direction. And as you can see, the sister chromatids are not identical due to that crossing over. Now we're back into anaphase, anaphase 2. The sister chromatids separate and move to opposite poles. So they will separate right in the middle. I'm not going to do this to all, but they will separate right in that middle and be pulled by the spindle fibers to opposite poles. Oops. This clay is not my friend tonight. So they separate and they pull towards those opposite poles. Okay, so that is anaphase 2. And then we are back in telophase 2 and cytokinesis. Now, with this, a new nuclei forms, and then the chromosomes uncoil, and then cytokinesis occurs. So during this, the cell actually will split again. The spindle fibers go away. And this is where we get our 4 haploid cells, as each of them have now created 2 more. So we have to make sure we have our 3 on this side, 3 on this side. And again, as you can tell, they are not identical. Each cell is going to be genetically unique. So as you can see, I have a blue and a pink, blue, blue, pink, blue, and then I have blue, 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 pink, 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 blue, pink, pink, blue, pink, blue, blue, blue. So as you can see there, each cell is genetically unique, as I already said. Another thing about difference about mitosis and meiosis is meiosis is going to be used for sexual reproduction, whereas mitosis is purely for growth and repair of the cells. And that is meiosis and mitosis.